Fresh cut fruits and vegetables usually respond differently than whole fruits and vegetables, and you'll see why during the course of this presentation. It's very important that you know the basic biology of whole and fresh cut commodities, and this is going to help you develop strategies to achieve quality retention during handling and marketing. Many of you know that already, but unexpected things will happen, and you're going to have to deal with them. So it's very important that you know the biology to solve those unexpected issues. Genetic modifications are happening, and scientists are working on that, and they can prevent some negative changes that make the plant. They can also make the plant more susceptible to stresses. Also, it's important to know when a specific fresh cut problem is a result of a physical, biochemical, and/or physiological change, and this is going to give you the opportunity to target treatments to enhance the beneficial effects and diminish the negative effects of wounding. So let's start with a random harvested crop. Let's say an apple. You all know that an apple, when you detach it from the plant, it's still a living organism. It doesn't die when you cut it. It still lives. So you have, like we do, metabolic changes happening in the apple. Carbohydrates are consumed, organic acids are produced, sugars, fats, and so on. And how they do that? They consume oxygen from the environment and they emit carbon dioxide. Believe it or not, they also emit heat. You cannot feel it because usually one apple is very little for us to feel it, but if you have a pile of grass which is decomposing, you have heat emitted if you put your hand in that pile of grass, for example. Water vapor through transpiration comes out of the fruit, and so does ethylene for most crops. And then, as Florence mentioned earlier, aroma is emitted from all crops, but it can also be absorbed. Let's say you store a load of apples with garlic, you're going to have really nice smelling apples after all. Ethylene is also absorbed, and it can change the metabolic processes, and we'll talk a little bit about that. So now let's go to this very easy plant cell diagram that I'll try to make it as easy as possible for you all. This is a very organized system. So it has a lot of compartments that I will show one by one, and each one carries out very specific functions. If you disrupt the whole structure, this is what we do when we cut the product, when we do fresh cut, it cannot function properly. So then you get all these problems that we talked about. So you have the cell membrane around it, and another number of membranes that protect the cell and compartmentalize it. What happens when you cut the cell? You have leaks. The cytoplasm is an amorphous substance where all the little organelles float in. Then you have the nucleus, which contains DNA, and it tells your cell how to run and what enzymes to make and what processes it has to do. Following, we have these little red dots that we call the mitochondria, and they're the powerhouse of the cell. They make energy that's required, and they keep the cell alive. They also help with respiration. Next, we have the Golgi complex and the endoplasmatic reticulum, and these are used for protein and carbohydrate processing. Following, you see the amyloplast, and that's where starch is being saved, and these exist in plants like potatoes, the tubers that store starch. They're colorless, and they are found in plant tissues that don't do photosynthesis. Then you have this big elephant in the room that's called the vacuole, and it's protected by another membrane. What are we missing? Color. Color is important. So chloroplasts are present in plants and plant parts that are green, and they can often be converted into other colors, and then we call them chromoplasts. You got to love these Greeks. They have a name for everything. So for tomato, for example, you have red color. And the pigment that gives the red color is called lycopene. And they are stored into these chromoplasts. Then you have here your cell wall. The cell wall is like fiberglass, imagine. It's composed of cellulose and microfibers, pectins, hemicelluloses that keep all these things together. The brown line around it is the middle lamella, and when it degrades, and this can happen, let's say, if you get a peach and you chill it, it changes its texture. So when you bite a chilled peach, you're gonna get a less juicy and mealy texture that we don't like. On the other hand, when you bite a fresh apple, you get this crunchy feeling, and it means that the lamella break nicely, and then you have the feeling in your teeth that you want. Moving on, let's look at the cross-section of a zucchini here. On the outer part, you have the cuticle, which is a waxy covering that protects the plant part. Why it is important to have a cuticle? It's protection, as I said. And when you cut it, you expose that plant to the air or the water or whatever the conditions are. The epidermis is under the cuticle, and they can create disorders like russeting in apples. You have vascular tissues. I don't know if it's visible here, which is like the plumbing of the plant, and they can respond when they're disrupted. A good example is avocados. If you put avocado in the fridge and then you take it out, you often see these streaks. These are vascular tissues that have been chilled, and they change color. Moving on, you have structural tissue to support the plant part, and air spaces, which are important to allow the air to move through the product. Now let's talk about gas exchange. As I told you, the plant parts are living organisms, so they emit gases and they absorb gases. But this doesn't happen from the same part of the plant in all crops. On the left we have our apple, and on the right we have a tomato. The stem scar for each crop is different. In the apple we have a very small stem scar, and in a tomato we have a big one. Apples are different than tomatoes because they have a different skin, and they have those little dots that we call lenticels. Most of the gas exchange in our apple is happening through the skin, through these lenticels, but on the other hand, in the tomato we have a very glossy appearance. The cuticle and the epidermis of the tomato is real hard, and they last for a long time. You can go to a field where tomatoes have been grown, and you can see these little, thin skins of tomato even a year later. So you have an impermeable skin that gases cannot penetrate. So in this case, 80 to 90% of the gas exchange in tomatoes happens through the stem scar. A good experiment that you can do is if you get a mature green tomato and you cover the stem scar with wax, your tomato will never ripen because there won't be gas exchange through this stem scar. So what you do when you remove the epidermal layer, you alter the internal gas mixture of the product. And this can have two opposite effects. First, it will inhibit the gas diffusion. If you put the product in water, this will inhibit the gas diffusion because gas moves much slower through the water than through the air. And secondly, it's going to enhance gas diffusion because this cut, unprotected surface will be exposed as the liquid is reabsorbed or evaporates. And I'm going to show you an example about that. So we have a fresh cut packaged product, you can call it fresh cut tomatoes. and you have a film that protects it. What happens after you cut and you package this product in there? Again, we make things simple. It's never so simple in nature. You have oxygen and sugar being consumed, and then you have carbon dioxide emitted. The volumes of the oxygen consumed and carbon dioxide produced are equal. However, the solubility of the oxygen is far less than the CO2, and the volume of the bag initially will decrease. So you have the bag the volume being decreased as the respiration converts oxygen into CO2. Since the CO2 is being absorbed by the tissue that's been just cut. Later, and that's called the aerobic respiration because you have oxygen in the equation. Later, we switch to anaerobic respiration. And you can see here there is no oxygen involved in this process. Oxygen declines and cells go into anaerobic. So they produce large amounts of CO2. Then what happens is something you all have seen. CO2 starts to accumulate in the bag and it'll start puffing. So you have, these bags of salad or whatever the product is that have high CO2 levels in there. So changing internal gas concentrations can produce unexpected effects. And a great example is a trial that one of our retired colleagues did, and I will show you some pictures of it. When you change the gas activity and the internal gas concentrations, you can stimulate responses that have been eliminated throughout the years through domestication and breeding. One of these examples is called viviparous, when the seeds can germinate while they're still in the fruit. So this colleague of ours had a great idea. We know that our restaurant industry likes sliced tomatoes, and we harvest our tomatoes green, mature green in California, and we ship them cross country. His idea was like, why not slice them here and then ripen them sliced and send them to Chicago or to New York red. So he took a mature green tomato and he sliced it. He was expecting that after a few days in the right conditions, you will get something like that. However, he altered the water activity and the internal gas composition, and that's what the result looked like. I don't know if it's visible to you, but you have tomato sprouts. I mean, 20 years ago, it was not popular, but maybe nowadays, we like sprouts, so maybe now it's a thing. So we can try it again. We'll see what we get. So let's summarize, and I want to stress it again that fresh cut, when you talk about fresh cut, you talk about wounding. And wounding can be from light and minimally processed, but it can also be very severe when you cut the product and you mince it into very small pieces. So the response of that plant part will depend on the type of the wound, as well as the previous or subsequent stresses. Let's say you had stresses in the field, or you have stresses in the retail chain. Plants have evolved a limited number of physiological responses to wounding, and I will show some of them. We can summarize the responses into three often overlapping ways. One of them is that they can respond as physical objects. Another one is they can respond as biochemical solutions when they react with some existing compounds. And last, they respond as physiologically and metabolically active tissues. And this includes the synthesis or activation of enzyme and accumulation of products. I'm going to show you a result about when plants respond to wounding as physical object. You all know baby carrots, right? You know how they're produced? Are we growing little tiny carrots and then you put them in bags? How do we produce them? Who knows? So we get a long carrot and we cut it into 3 or 4 pieces, and then we put these little carrots in like a dryer, imagine, and then it shaves off the sides of it and it makes it look like a tiny baby carrot. I will admit, Maybe 10 years ago, I didn't know that it existed. I was a young man. I thought it was tiny carrots that are washed and put in bags. I'll admit that. So when we do that and we produce a small carrot, we disrupt the cell walls, and you expose the plant part into the outside atmosphere. So what you have, and you often see that, is you have evaporation in the bag when you open it, and you have water loss. So your carrot in the factory looked like that, but then when you store it in your refrigerator for a week or 10 days and the bag is cut open, you have this drying appearing. This is not dangerous. If you go online, people will write and rage about how this can kill you. It will not. If you put it back in water, it will rehydrate. But it's interesting to see, and you can sometimes see it with your bare eye, that you have remnants of cell walls sticking out. And that's the cells are dehydrated, and then you have this appearance. So maintaining turgor is very important to maintaining texture and product appeal. As solvents move across the cells across their membranes using osmosis. I'm not going to get too much into that because we don't have much time. I'm going to give another example when plants respond to wounding as biochemical solution. And an example of this is oxidation of some compounds that are called phenolics, which exist in plants, and they turn into pink or brown accordingly. Again, I'm not going to get into the chemistry and how this happens. You can read that at your own leisure. I'm going to give you some examples. We have here 5 different crops. On the left, tomato, celery, zucchini, apple, and banana. And we cut them at time 0, and we put them on the counter of our laboratory. And then we take pictures of them periodically, and you can see here that an hour later, our tomatoes and celery sticks look fine. The zucchini started changing color, but the banana and the apple have really visible changes. And as time progresses, you see how significantly different the results are between the bananas and the apples and the tomatoes. So if we could categorize those 3 groups of commodities. On the left, you have minimal browning. In the center, you have some slight browning. And on the right, bananas and apples have rapid and extensive browning. And this can be attributed to the differences in phenolic compounds and the phenolic synthesis that occur in the plant part. On the left, you have minimal phenolic synthesis. Zucchini have slight phenolic synthesis, and bananas and apples have high amounts of phenolics. So that is to say that not all tissues respond the same to wound-induced tissue discoloration. The tissues that brown rapidly have high levels of phenolics, low levels of antioxidants. Antioxidants are natural, and they can also be added to the fresh cut product, and what they do is they act against oxidation, which is what's happening to these plant parts. There are some enzymes like polyphenol oxidase and peroxide dismutase that occur, and they have also leaky membranes. On the other hand, tissues that brown slowly, like tomatoes, they have low level of phenolic compounds, high level of antioxidants, low levels of PPO and POD activity, and intact membranes. So if you want to prevent browning in these 2 tissue types, you got to employ different strategies. On the left, you want to reduce the level of phenolic compounds, either by genetic selection or by using different cultural practices. You can supply externally antioxidants and exclude oxygen from the packaging. You can reduce PPO and POD activity and increase the stability of those membranes. On the right, you prevent phenolic accumulation by reducing the wound signal and enzyme and phenolic synthesis. You can increase membrane stability and, in the end, exclude oxygen. Another example that's been around for a few years now is biotechnology that can deal with browning. And great example is apple. There's this new variety, the Arctic apple, that has been designed so that it doesn't express 4 PPO genes. And you see an example of a Granny Smith apple. The left is the Arctic variety, the Arctic cultivar, and the right is the regular one. And here you see a test where they produced apple juice and left it for 24 hours at room temperature. You see the differences are really big. Moving on, let's talk about an example when plants respond to wounding as physical object and at the same time as physiologically active tissues. And I will not get again into the background. I will show you an example of the respiration rate of a mature green tomato fruit. On the x-axis you have hours after wounding. The minus is before wounding, and the plus is after wounding, and then on the y you have milliliters of CO2 per kilogram hour. So the normal non-wounded rate of respiration of a tomato, a mature green tomato, is around 12 milliliters of CO2 per kilogram hour. When you wound, when you cut your fruit in eighths times 0, and you measure the CO2 levels, you see this big spike of respiration. So you have a 3.8-fold increase from 12 to 45 milliliters per CO2 per kilogram hour. However, you would think that this all is because of the wound enhanced respiration. If you take a closer look, in the beginning you get the CO2 which was dissolved in the tissue escaping in the atmosphere, and that's the initial CO2 burst that you see, that you measure, and then later, this is the actual wound-enhanced respiration that happens a little later, 0.2 hours after cutting your fruit in eighths, you get this. So the total is this, but it's not all because you injured your fruit. Moving on, let's talk about another example of respiration of intact and shredded lettuce. This study was done by our colleague who you will hear from, Marita Cantwell, where she stored lettuce heads in 4 different temperatures, 2.5, 5, 7.5, and 10 degrees Celsius. So you can see that, of course, as we know as produce professionals, the lower the temperature for leafy greens, the lower the respiration rates. And then, but when you shred your lettuce and you measure again those respiration rates, you see that you have a significant increases in those respiration rates. So summarizing, shredding increases greatly the respiration rate. Controlling the temperature is extremely important. The lower the temperature, the lower the respiration rate. And something that's interesting to look at is that the intact lettuce at 10 degrees expires about the same as the shredded lettuce at 2.5. So you can see the damage we're making to the product and how we make it expire more by shredding it. Here's the, we're going to talk a little bit about climacteric and non-climacteric fruit. Florence mentioned about it. When we talk, non-climacteric fruit are the immature green fruit, vegetative tissues, and fruit like strawberries, pineapples that do not ripen on their own after harvest. So what you have is, usually these are the equilibrium, ethylene, and carbon dioxide production, and after wounding, after treatment, you have an increase of the carbon dioxide and the ethylene levels, and then they go back to close to normal. On the other hand, when you have climacteric fruits, like apples and bananas and plants that produce a lot of ethylene and they're sensitive to ethylene, this is how the ethylene production rates look like naturally. So, and the carbon dioxide level also, the rise coincides with the ethylene spike. So when you wound your product there, this is what you get. So you see that wounding causes an early onset of the climacteric. So ripening occurs more rapidly. So senescence also occurs more rapidly. So your product dies shorter, it has a shorter shelf life. In this case, managing your ethylene levels is very important. Let's move on. I'm going to skip that because I don't have much time. Ethylene should be minimized to maintain quality. And low levels of ethylene can promote ripening of climacteric fruits and vegetables. Even very tiny amounts of ethylene, like 0.02 microliters per liter, can adversely affect the storage life of non-climacteric vegetables. I'm going to skip this as well, I'm sorry, but we're running late. And I'm going to talk about how to control the effects of ethylene. The question that we have is should we remove the ethylene from the atmosphere, or we should make the tissue less sensitive to ethylene? Often when we talk about fresh cuts, we talk about controlled or modified atmosphere. So ventilation is not always easy. It's often impractical. A beneficial effect of a traditional controlled atmosphere is that low oxygen will reduce the ethylene synthesis, and low oxygen, high CO2 levels reduce the sensitivity of the tissue to ethylene. Pre-storage treatments such as heat shock and 1-MCP can render the commodity less sensitive to ethylene, but the most important way of managing ethylene is temperature, temperature, temperature. So don't forget about that when you produce fresh cut products. Generally in post-harvest, our number one way of reducing the post-harvest losses is by managing the temperature appropriately. This is the last example I have, is the phenolic metabolism that leads to tissue discoloration, and you've all seen that in your lettuce. It doesn't happen immediately, it happens later as time progresses, and we've shown that when you make extra wounding, extra cuts, you have more of this russet coating on these crops. How you control that is you want to minimize stress before and after harvest. You want to use sharp knives during processing. You market your product as quickly as possible. You store and ship at 0 degrees Celsius for lettuce, and when you use CA or MA, you use packaging that excludes oxygen, and you increase the carbon dioxide levels. And with this, I'm going to come up to my first slide, which is the conclusions. You can go through it, and I'll be happy to take questions, one question, or maybe you can find me later during the day. I'll be here the whole day. So thank you.